Okay, so we're back again. We're talking about translation. And in order for cells to make proteins, the DNA code must be transcribed to mRNA. Because remember, the DNA is in the nucleus, it cannot leave the nucleus. Um, it is in that nucleus, and it is short. Bleh, it is protected by that nucleus. It's think of it as too big to leave also. And mRNA makes a copy of the DNA using complementary base pairs and carries the code from the nucleus to the ribosomes. So here we have RNA polymerase, and it makes, it's actually going to come through and it's going to build complementary nucleotides onto the DNA sequence. Now it's going to build complementary nucleotides, meaning that um, A is going to bond with U, and T is going to bind with C. Okay? Every time you would have seen a T in here binding, you now see a U. That mRNA is actually this single strand um, that is made from the DNA and is carried out to the ribosome. The ribosome actually assembles the protein using the code on the mRNA. So at the ribosome, amino acids are linked together to form specific proteins. So after that messenger RNA is formed, and it's formed from RNA polymerase, at the ribosome, amino acids are linked together to form specific proteins, okay? The amino acid sequence is directed by the mRNA molecule. So here we have AUG, and this is the mRNA sequence, and it's comes and it binds to the ribosome. And here we have tRNA. tRNA is going to come down and it's going to bind to mRNA. The tRNA is going to bring with it the amino acid. Okay, so here we have tRNA, and like I said before, AUG is going to start every codon. So here we have started with AUG. Here we have tRNA has the anticodon coming down and it's binding to the codon, and that anticodon is complementary to the codon, and that's A binds with U, U binds with A, G binds with C, and it brings with it a amino acid, a specific amino acid that this codon down here codes for. Now don't get that confused when you're looking up your, because I'm going to show you here in a minute how to look up specific amino acids. Don't confuse your mRNA, your codon, with your anticodon. The codon is actually what codes for the amino acid. Here we just have an illustration, and here we have tRNA. It's bringing down MET and binds to AUG. Here we have another site, the A site, where another tRNA comes and binds to the codon. The anticodon comes and binds to the codon, brings with it an amino acid. When the two are side by side, a polypeptide bond, or I'm sorry, a peptide bond is going to form. Once that peptide bond has formed, the empty tRNA can leave, and another amino acid can come and bind to another codon. And here we have the protein actually forming, the proteins actually being assembled, and peptide bonds are forming between them. Okay, so we're going to go through this. Hold on just. Okay, so now we're back, and we have. Hold on just a second. We have a DNA sequence, and it's TAC, TT, TTG, TTC, and you guys can read. Oops, I made a mistake here. Can anybody point it out to me? Yep, that's right. I put a U in here and this is DNA. Let me change that. Okay, now we're back and we have the correct nitrogen base in here. We have thymine instead of uracil. Now what I want to do is I want to build the correct mRNA sequence, and while you're at home I want you to do this on your own. Just try to do it on your own and see if you can build the complementary base pairs. So T is going to bind with A is going to bind with U because we're translating it to mRNA, and where we would have seen a T we now see a U, so A is going to bind with U. C is going to bind with G. T is going to bond with A's, T is going to bond with A again. T is going to bond with A again. So I'm going to give you like 30 seconds to do this on your own, and I'll show it. Okay, so here we have AUG, AA, AAC, AAG, GUA, UAG. And that should be your mRNA sequence right now. Check your work. If you got it right, give yourself a pat on the back. If you got it wrong, don't beat yourself up. Email me a question that you might have, or maybe just rewind the video. Okay, moving back, mRNA. mRNA sequence, we have, again, the same one that we just had before. And now we're going to make a tRNA sequence. Let me just make sure, check my work here. Yeah. We're going to make a tRNA sequence here. So now I want you guys to try at home to make a tRNA sequence, and this is just going to be complementary to the RNA sequence. A is going to bind with U because we're still in RNA. U is going to bind with A. A is going to bind with or G is going to bind with A. A is going to bind with T. Sorry, not T, because we're in RNA. U, A, U, A, U. And I'll let you finish that up. Okay, if you got UAC, UU, UUG, UUC, CAU, AUC, then give yourselves a pat on the back. If not, email me a question or rewind the video. Moving on. Here is what we call a codon chart, and there are so many different codon charts out there, and this one's probably the most complicated, so I'm going to show you guys today how to read this codon chart. Well, if you'll remember, we use a codon chart to look up amino acids, and we'll use a codon, which is found on the mRNA, not the tRNA. Don't go for the tRNA, go for the mRNA, and we'll look up each of the mRNA codons on the codon chart. So we have AUG to begin with. AUG. The first base in AUG we find on these within these brackets. So the first one is A. So this is our first bracket. So we're going to look within this bracket right here. Within this bracket, A. Our second base, you'll see the title up here, second base. We look within this column because we look for U. So we look within this column, within this bracket. So A, U, and so we know it's within this column of this bracket. And then our third base, we look over here and we find G, okay? So, whoops. So we find A within this column, A within this bracket, U within this column, and then we look for our third brace here, and we find G. AUG codes for met. So our first amino acid is met. And that's right. Our second amino acid is lys. LYS. Our second amino acid is lys. LYS. Or let's look it up on the codon chart. It's AAA. So we're looking for the first base, and again, it's an A. So we're within this bracket. Our second base is going to be another A. So we know that we're within this row. Within this column. So A within this bracket in this row. And then on the third base, we look up A again. So it's within this row. So it's lys. LYS. And again, we're right. Now I'm going to try AAC. A. So we're going to go first base. So out of these, our first base is A. So again, we're in this bracket. Then our second base is A again. And so it's within this column. So we're within this column of this bracket. So we're looking at these four right here. Then our third base is going to be right here. So, and I can't remember, was it? Let's see. C. So it's ASN. And that's correct. So I urge you guys to do that on your own. Maybe pause the video and see if you can look up all these different amino acids from the sequence, and if you cannot, if there's a mistake, let me know. But if you cannot, just email me a question or maybe rewind the video. This is a very complicated chart to read, and I am going to show you several different charts that are less complicated, but I want you to be able to read this just in case, on the off chance they want to give you a really hard question on your EOC. Okay, so ending today, you should know what are the three differences between DNA and RNA. You should know what are the three types of RNA and what their functions are. And then you should be able to describe the two steps in protein synthesis.